Okay, guys, let's start. My name is Cvetan Popov. I'm going to be your tutor for the course of histology, cytology, and embryology. Today, we're going to do the first topic from the last week, the one which we missed. Starting with microscopes, we're going to talk a little bit about different microscopes. We're also going to talk about how to prepare permanent histological slides. What is cells, what is tissues, and we're going to talk about external cell morphology. First of all, before we start, it's good to get introduced to the cells, to the cell biology, to tissues, to the histology, and therefore I put two slides in order to explain what is cells. So, as we all know, cell is the basic structural and functional unit of life. What makes the cells the basic structure and functional unit of life is the ability of producing energy. So very important one, cells are able to produce their own energy. Then, what else they are able to produce? They're able to produce the building blocks, proteins, lipids, and carbohydrates they need to use in order to form the cells or to maintain their life. And also, very importantly, guys, cells can self-reproduce. This is very important one because this means that one cell can produce a copy of itself 2 times. So that's multiplying. Right. Cell biology is the science which actually look for the different cell-cell interactions, about the self-behavior, self-morphology, and everything else. Is the cell biology. But we know that organisms, guys, can be built of simple cell, these are unicellular organisms, or of multiple cells, those are multiple cellular organisms. Humans are multicellular organisms. This means that the cells which forms them, guys, they tend to group and form some specific structures inside the organism, which we call tissues. Tissues can be 4 different types. This one is going to be our subject on histology. But before that, the science which study the tissues, guys, is called histology. Here on this presentation, you can go in to see several different examples of tissue slides, guys. Here, for example, we're looking for the glandular epithelium. This one here is called covering epithelium. This is a fat tissue. And here you're going to see the oocyte within the follicle, which localized in the ovary, guys. So you're going to see a lot of examples of histological tissue. Now, these structures we observe thanks to a special tools we call microscopes. Microscopes can be several different types, but we're going to hardly separate them in 2. The one which use a white light as a source and the one which use electrons as a source of light. So the light microscopes, guys, can be subdivided into 3 categories. Normal light microscopes, as you're going to see them in the left here, this one. Those are typical ones. We're going to use them a lot in our practice, guys. And those are the one which has the ability to use the normal light, the sunlight, if you want, the one which has the full spectrum of the light, and to observe the histological slides. So there you're going to see that the histological slides which are stained, and we see them with our eyes that they are stained pink and purple. For example, under the microscope will be also pink and purple. The other 2 microscopes, guys, use a little bit more highly intense light, for example, fluorescent light. This is also white light, but very strong light, intensive light, so you are not able to directly see inside the light. Therefore, you use a special filters in order to sort a specific length to pass through. And this microscope, especially the fluorescent, and also this is typical for the laser microscopes, they use fluorochromes. Those are chemicals which actually are able to get excited in a specific wavelength and then emit a light with a specific wavelength. For example, they absorb the light below 488, but they emit a light at 488. This means the green light. We have fluorochromes which can emit red, white, yellow, orange, blue, violet, or red color. So a lot of light. So all the spectrum of the rainbow, guys, you can imagine there, it can emit it. And then you can sort it out, and then you can analyze only this part. So compared to these two microscopes, guys, confocal laser microscopes, which are this one, you're going to see they're pretty big. They're actually unique because they don't use the light as a source, but they use a specific reference. So this means they use lasers. Lasers, by definition, are light with a specific length. So, for example, you have blue light, green light, and red light. Those are all lasers, which specific length. For example, I think the green one is between 488 and 489. The red one, I think, is between 545 and 547. I think in this diapason of nanometers. So this means that the fluorescent molecules, they need to be precisely this one in order to see them under the microscope. Okay, this is about the light microscopes, guys. The structure of the light microscopes is pretty important, this one, because we are going to use these microscopes extensively in our study here. So what you're going to see here, this is the common structure of the light microscope. It consists of two parts. I'm going to surround them now. This part here is called mechanical part. And this part here and here are optical parts, guys. So, mechanical part, as you're going to see, consists of arm or stage, and also we have the base on which one the whole microscope lies. On the base one, it mounts the camera, the light source here. The light source is switched on by this button here. When the light is too bright or too extensive, you can decrease it, or you can increase it if it's too dim. So the light comes from here and goes through this structure we call the condenser onto the table. This is also called mechanical stage also. This one here is very important because here we put our histological slides here and observe them. So when we put the histological slide, you hold it thanks to this holder here. There is a small holder, which is here. Let me see if I can clean this one a little bit. So that's, okay, now it's not overloaded. So this holder, guys, here, this one here, it's actually the one which will lock your slide, and then you can use these two structures. This is called stage controllers in order to move the slides up, forward, and backward, like this and this way, or this and this way. So this way and this way. So left and right, forward and backward. This is very important for us because this will allow us to guide ourselves to the slide very precisely. It's a narrow, very narrow way to find our whatever we want to see. So, but in order to focus the slides, we need to use these coarse adjustments. This one here. This is very important because this moves the table up and down, in order to find the focus. Focus, guys, means that you need to have a sharp image. Let me show you how sharp it's supposed to be, this image. It's supposed to be this sharp, as this one. We want to see a very defined borders of the structures, guys. So therefore, we can use these coarse adjustments. With the hard one or coarse adjustment tool here, you're going to see you can scroll it up and down until you don't see the shadows, or actually you see the outline of the tissue. But then you use the fine adjuster, or I call it fine tuner, in order to precisely focus the picture you share the sharpest image of separation is possible. Then, after we do these things, we see the stuff, what we do is we use the optical parts here, guys. Optical parts includes these structures here. Those structures here are called objectives. Objectives can vary in size and magnification, guys. For example, there are very short objectives which are kind of small magnification. They have very simple lenses. There's a very small magnifications. This is between 4.5 to 5 times magnification. Then we can observe another objective which can increase the image even more, and also, for example, the 10 times magnifications. Then we have 20 times, eventually 40 times, 63 times, and the bigger one which we have on the microscopes, it's 100 times magnification. 100 times magnification, and some of the 63 times magnifications are special ones because they require a special solutions we call the mineral resins in order to function properly. Therefore, we use them very hardly. So from just very limited access to these objectives. Most of the time, we're going to use the smaller magnifications, guys, because there will be enough information on them in order to see whatever you want to see. Now, our microscopes, guys, has 4 different types of magnification. We have 4-time magnification. We have 10 times magnification. We have 40 times magnification. And as I said before, 100 times magnification, which we don't going to use, guys. So we're going to limit ourselves up to 40 times magnification. After that, the light comes from here, goes to the objectives and goes to the oculars here. So what's happened here? You put your eyes, both of your eyes here, guys. Those are the called oculars or eyepieces. Here we are going to set up the microscopes the way that you need to do it in order to see a unique for your image. So this means that you need to move these lenses in order to find your perfect balance or distance between the eyes, so it doesn't push them too much, guys. So this is in general the structure of the light microscope, the one we're going to use a lot with you when you come here eventually soon, I hope. Now, continue on. Let's say some words about the electron microscopes, guys. So here I give you 3 examples of electron microscopes. This one is the old timer, transitional electron microscope. Guys, you're going to see pretty big machine. It requires its own room in order to function, guys. This one is the improved version of that, if you want to put it that way. So this is the improved version of the transmission electron microscope. What is the improved one? The size. You see, this is not so big in size. These are also called benchtop microscopes, guys. So they can fit in one benchtop. No problem. Magnification are almost the same, even though this one is a little bit more. I think this can go up to 2.3 times. 2.3 million times magnification, guys. So it's not exactly the same. But anyway, both of the microscopes though use a very high voltages, guys. Those are with the kilovolts ranges, so they're very expensive to use. That's why we usually don't do it too often. The other one here, which I put you as an example, is a new development which actually use low voltages to do the same thing. So they're kind of a little bit cheaper to use during the practical parts, guys. So maybe if you're lucky, you can use such things in your practical parts. Now, continue on. The structure, guys, and the function of the microscope is pretty similar. Here, for example, you see a light microscope, and here is the electron microscope. The source of light is different, of course. This is a light source. This is the electron source. And depending on that one, guys, the lenses are different. Here, for example, we have optical lenses, which are normally crystals or glass lens and stuff like that. Compared to them, the lenses here are magnets, guys. Magnets, which can be positively and or negatively charged. They're actually electromagnets, so they can switch. They can be now negative, but after that can become a positive and stuff like that. Depends what you want to do. Now, here is the objectives, guys. Here we put them also, and here is the way of detection. Nowadays, no one usually look under the microscope, just for students, guys. Most of the time you do detections by cameras. This is how it's observed both of the slides. So you see the structure and the function, it's pretty similar between the both microscopes, guys. Now, we use microscopes in order to analyze the histological slides, guys. This is why they invented. Histological slides can be kind of separate into main categories. We talk about cell slides and we talk about tissue slides. What's the difference? Cell slides, you're going to see those are cells which we can culture on the slides. This means that we can put them on and then they can get to the specific density, or actually and the amount is enough so for you to then do experiments with them, guys. Tissues are different. Tissues, this is a histological tissues, like in organs, for example, they can become a histological slides, tissue slides. Now, starting first with the cell slides, guys. As I said before, those are cells which you can culture. Examples of such cells are stem cells, for example, stem cells, cancer cells, lymphocytes, guys, hematopoietic stem cells. Those are all examples we can actually culture. So what do? You plate your cells on the slide, then you culture them to the specific density you get them. Then what you do is fixation. Fixation is a key method here. What is fixation doing, guys? It kills the cells, but keep them in kind of closer to the normal shape. So we don't introduce artifacts when you do the fixation process. As a fixative, you can use several different categories of fixative. For example, physical agents can be heat or microwaves. Those are very potent fixatives, guys. Also, aldehydes, 4-aldehyde, formaldehyde, poroformaldehyde, guys, butylaldehyde, those are typical fixatives, typical for the histology, though. For cells, most likely, we do these coagulants like methanol, ethanol, acetone, acetic acid, guys. These are typical for the cells. Now, so compared to the cell slides, guys, tissue slides, as I said before, they are three-dimensional. They have volume. Those are, you can imagine, organs. When I talk about tissue slides, when I close my eyes, I can imagine kidney. You know how the kidney looks like. So the kidneys, guys, are three-dimensional. In order to have them inside the, if you want to make slices of them, you need to put them in some stuff which we call carriers. And the carriers can be two different types. Most likely. We have a paraffin carriers. This is the commonly used one, and you're going to see here several different slices included in the paraffin. As you're going to see here, I don't know what is this. But anyway, we can have a bone, we can have some kidney, as I said before, you can have brain, whatever inside. But the paraffin has also some limitations. What are the limitations? Since the paraffin needs to be heated in order to melt and then to put the tissue on, some of the very, how to say, loose tissues, like fat, for example, which is filled with lipids, when you put it in, the heat will melt the lipids and they will go out of the tissues. So you're going to see an empty cells there, which is not optimal for us. Therefore, we can use, for example, the cryomedias in order to put them in and then observe them. Okay. So histological slides, guys, how they produced. Here we're going to see the whole scheme of the production of the histological slides, starting with the first step, minimization. When you get the whole organ, for example, kidney, you don't do the full organ histology, guys. Usually you take some small parts of it. So this means that you minimize the amount, because if you have the whole organ, it's going to be difficult to fix it properly. Then following the other steps, and of course also for the cutting, because you don't have such a big slides to put the whole organs on. So that's why first of all we do minimization. So then we have the fixation process. I told you fixation is very important because this one kills the cells. This means stops all the metabolic stuff which happens inside the cells, but they remain as close as possible to the original one, to the intact one. And after that, after you fix the tissue, what do? You do the dehydration. Dehydration process is an essential process here because it removes the water from the tissue. There are several reasons for that. First of all, water, guys, is a very good source of fungi and bacteria grow inside. So this is a very good source. Dead tissue is the best source of growing fungi and also bacteria. Also, if you have the water inside the tissue, you can hardly put the paraffin in. You know what's happened when you put the oil on top of the water? They don't mix. Apparently, they stay in two different lanes, or they just different. They separate kind of layers of stuff. So therefore, we need to do the dehydration. Dehydration process, guys, involves using alcohols, or we call it also alcohol rows, starting with the lower concentration between 70 to 80%, then 90, then several times 100% until we are sure that the whole water goes out of the tissue. So then what's happened? Since the water goes out of the tissue, and most of the tissues, you know, they are formed of 90% water, the cells. So the cells a little bit shrink inside. So in order to get them back to the original size, you introduce the second stage, or actually the third stage, which is called clearing. Clearing process, guys, is the one you put something like a high weight alcohols and also some of the mineral resins, which actually goes inside and fill the gaps which the water remain left after it took out. So after the cleaning process, guys, clearing process, we put the paraffin or the krill media, doesn't matter, one of it. If it's a paraffin, you see it's melted paraffin. It's between 52 and 60 degrees. The freezing media is liquid one at room temperature, but when you put it in cold conditions, it's froze very efficiently, but it's very greasy, very rich of fatty stuff. So it doesn't kind of damage the tissue. So this one, when it goes in, it kicks out the alcohol, like xylene, xylol, or the mineral resins, guys, fits inside, and then you have your tissue embedded in the carrier. After which one, you're going to start sectioning it or cutting the tissues, guys. When you cut the tissues, you're going to observe kind of whitish structures there, but we will not see anything like cell or tissues there. It's hardly visible anything, just some white structures. So in order to visualize the cells and especially to see the difference between the cells and the tissues, we do stainings. Stainings can be several different types. Of course, more common one are the chemical stainings. Here I give you an example of a common chemical stainings you can observe under the microscope. You're going to see a wide variety of stainings. Keep in mind that what I show you here is not even 5% of all chemical stainings can be done. In our studies, guys, we're going to see several of these stainings. For example, this one, cresol violet, we're going to see a pulse reaction, we're going to see hematoxylin, which is not even here included, and stuff like this. Silver impregnation is these things, doesn't have them here. But we have a reticulin staining, which is you're going to see a very interesting one. It's reticular fibers tends to bind to the silver ions very good. Now, okay, so those are the chemical stainings. But as I said before, chemicals can be very huge amount of stuff, and actually you can narrow down a specific staining in order to see a specific thing inside the cells. Example, here I have the same tissue twice. This is epithelium layer of the small intestine on the left and on the right, guys. But one is stained with hematoxylin, the other one is stained with a pulse reaction. Hematoxylin, guys, is a very common, I mean, the most commonly used staining in histology. It actually stains the nucleus, this one, with a purple, and then the whole cell, protein cell, stains with a pink color. This is the eosin. Now, when we stain with hematoxylin eosin, you're going to see the structure of the tissue, but we're going to observe also cells which are not stained. This one, we put them G on them. Those are the goblet cells, guys, or also called glandular cells. Those cells are filled with mucopolysaccharides or glucosaminoglycans. Those are typical for them, guys. So they will not be stained with hematoxylinogen. So we're going to see them as a holes, empty space there. But if I use the pass reaction, as I said before, you're going to see this one very intensively stained compared to the rest of the tissue, guys. So therefore, if I want to observe just the goblet cells, then I can do these stainings. But if I want to see the whole tissue, I will not do the pass staining, I will do the hematoxylinogen staining, guys. So that's why when you want to see something, you can easily narrow down the staining you can use for. So the chemical staining is not the only thing you can do into the histology. You can also use antibodies. And the definition, as you know, antibodies are proteins which are produced against specific antigens. Antigens can vary a lot. It can be a receptor part of the cells, it can be part of the whole cells, or it can be the whole cells in general. You can also have an antibody against the whole cells. Normally, when the antibodies are recognizing part of the cells, those are called primary antibodies, guys. And actually what you can do, they go and attach. You can label the primary antibodies if the receptor or whatever part is targeting is very dominant in the cells. So you can label them with some fluorochromes, the one which you use under the fluorescent microscope, and then you can observe it. But there is also an exception. If the receptors or the part of the cells you want to target is not so pronounced, then you can use a secondary antibodies, as the presence of secondary antibodies, guys, amplifies the signal, as you're going to see here, since they bind to the primary antibody. So this is the one. Carry out different fluorochromes, you're going to see a stronger signal, even though your receptor or whatever you're targeting there, it's in a very small amount. Okay. This is typical, though, staining for the cells, because you can have no interference from the tissue. But when you talk about human tissues, guys, especially, under the fluorescent light, you can just put the whole tissue not stained with anything under fluorescent light. You're going to see they're pretty green. So this means that human tissue is very autofluorescent, not only humans, most of the species which actually consume meat. So they are pretty autofluorescent, guys. And even though you don't eat meat, you still have autofluorescent tissue. So therefore, for histology, normally we use antibodies which are bound, the secondaries are bound to specific structures which we call enzymes in this respect, peroxidases. You're going to see we have also another type of structures, but we don't care. So in this picture, we are looking for the lysozyme, guys. This is an enzyme which is located here in this glandular epithelium. So we target it with the primary antibody, with the secondary antibody which has the peroxidase. We actually attach to the primary, and then what we do, we give a mixture of substrate. Half of the substrate, guys, is a peroxide, hydrogen peroxide. Hydrogen peroxide. Yeah, I draw very good. So hydrogen peroxide, guys, here it's going to be destroyed by this peroxidase to water and oxygen. This we call the reactive oxygen, guys, since this oxygen can go and attach very actively to other parts of the structures. The other half of it, so stuff which we call DAB or diaminobenzidine. This is a special chemical, guys, which actually can attach this oxygen piece, this reactive oxygen can attach very dominantly to this kind of structure, and then it changes the colors. So normally it's kind of transparent, as you're going to see it here, but when it has an oxygen, it turns to kind of brownish color. So here, for example, here and here, we're going to observe the presence of the lysozyme, because this is the reaction we're going to see together with you. Now, this is about antibodies. There is one more way to stain the slides, guys. The one way is called quiche or fish. This is actually a very interesting way of staining it, because you can see even very small changes. Fish and quiche is done with probes. Those probes are called DNA probes. They actually targeting specific RNA or DNA molecules as they're labeled. When they're labeled, you're going to see when they're attached to it, they're going to have a color. For example, on this one here, you're going to see HER2 receptor stain. You're going to see it here in the center. These dots, big, very big dots, guys, those are the presence of HER2 receptor. On the fish one also, this is the fluorescent, this is a colorimetrical, this is a fluorescent one. Here is with red. These areas which are red, guys, this means that you have the presence of these probes there, of this gene there. So what is HER2, guys? Why do I put so much effort on that one? HER2, guys, is supposed to be, actually this is not supposed to be, but it is a hallmark of the breast carcinoma. This is later stages the breast carcinoma gets, the expression of the HER2s gets more pronounced, guys. That's why it's linked between, there is a link between the presence and the breast carcinoma here. Now, this is about a small introduction I prepared for you for the different histological stuff. Now, let's go to the topic for today, which is actually cell morphology. First, we start with the cell shape. When we talk about cell morphology, the first thing I always imagine is the cell shape. Well, the cell shape, guys, as you're going to see here, can vary a lot. It can be a bipolar, it can be multipolar, it can be circular, it can be kind of unique, like the sperm here and stuff like that. In general, guys, the cell shape depends on a lot of things, but I narrow down here some interesting ones. For example, function, very important one. If your muscle cells looks like this or like this, when it's contracted, it can contract this way, this way, or that way. So how the cell should know which way to contract? Well, it doesn't. And when it contracts, most likely will contract the whole thing. So this means that you're going to see a very random contractions, which can do whatever. And you're going to see a very efficient muscle contraction, which actually may cost your life, guys. So therefore, nature is very selective what you do, and then builds the muscle cells looks like this. They usually bipolar or kind of bipolar. We're going to see that there are some differences, but most likely they tend to be kind of bipolar. So they have a limited contraction away. Neurons, guys, another example typical for the cell shape. You know that neurons are normally multipolar. They have several protrusions, as most pronounced is actually the axon, this very long one, and then we have here the dendrites. If your neuron looks like this, for example, ovular shape, so first of all, you will lose the ability of neuron to transmit the neuronal impulse to a very long distances. This is first thing you're going to lose. The second thing you're going to lose is the connection between the cells. One neuron normally connects to around 16,000 other neurons, guys. Well, in this respect, this one will connect to a maximum hundreds. Let's say several hundred cells. Be generous, guys, but not more. So you see the difference between thousands and hundreds. So it's a lot. So therefore, neurons has this kind of shape in order to have even more connections. Interactions, guys, are also important for the cell shape. If you have cells which, if for example, this is your space, and if you put several different cells, guys, let's say 3, they will look like this, yeah? But if I put not 3 cells in the same space, guys, but I put, let's say, 30 cells, so they become something like this. Yeah, they will try to fit in, try to keep the volume, guys, but you're going to see that they're actually going in highness. They go high. This is what's happened. But if I put not 30, but I put, for example, 300 cells in the same space, you're going to see they become like these very big one. Cells, guys, they hardly attach to the bottom, hardly attach, but they, since they want to keep the maintain the volume, they kind of looks like this. So in the histology, we're going to teach about these things. Those are kind of different types of epithelium we're going to observe. The one is called squamous, cuboidal, and then comes to the columnar epithelium, guys. Now, so you see that interactions between the cells also very important one. Cytoskeleton, the other thing actually which is pretty important one, we're going to discuss with you when we get into the practical number 5, which is actually all about cytoskeleton and how structured it is. Now, continue on. Despite of cell shape, we have also, as a morphology, cell size. As I said before, the volume which the cells occupies, guys. Normally, cells fit in this medium range of 10 to 20 micrometers, guys, normally. But there are some small cells, which you're going to see here, which are less than 10 micrometers. Such cells are sperm cells, astroclea, erythrocytes. There's also cells which are bigger than 20 micrometer here, this one. The 20 micrometer, for example, oocytes, pyramidal cells of bad guys, those are big cells. Those, especially oocytes, can get to 130, 100 something micrometers. They're pretty big one. You're going to see them with you today also. Now, we've covered the cell shape and the cell size. Now we need to talk a little bit about the cell color. As I said before, most of the cells have no color, guys. No. So this is, for example, a slide which it's just recently cut. It's not stained yet. So what do you see here? So I purposely see here, I personally see here these blue lines. These blue lines, guys, here are not cells. Those are usually reflection of the connective tissue. You don't actually see it too, because it's actually pretty much thicker than that one. So I can tell you this slide maybe come from the kidney or from the eyes or from the heart, and you cannot justify that one because we have no proof. So I can be correct, but I can lie as well. So therefore, you know that cells are not stained in general. So that's why we need to introduce the staining process here. But there are some cells, for example, erythrocytes, melanocytes, fat cells, or muscle cells, which are stained initially, so you don't need to look for the staining for that, guys, even though we will still stain them. Anyway, okay, so with that one, we finish up with a small introduction, and we will go to the second stage, which we're going to see 4 different folders like this next to each other. One is called Cytology for English, the other one is Anatomy, then you have here the Cytology for Bulgarians, and then the other one is called Pathology. We are going to click on the Cytology for English learning, and then you're going to see all these folders inside. These numbers, guys, indicate the practical part we are with you now. So we're going to usually click on practical number 1, and when you click in, you're going to see all the histological slides. You can do it actually at the moment since I'm talking to you guys. Now, do you want me to go back to show you again the server here, this one? Should I keep it for a little bit longer in order to copy that one, or you don't need to? Just type here in the chat, so I will wait for you guys to tell me. I think you can take a print screens from this presentation, so you can, okay, good. So if you have it already, very good. So try to access it. Let me know if you can do it, guys. Now, continue on. We start first with the first slide, guys. It's called ovarium or ovary. As you all know, or you may not know, but the ovary is a paired organ, guys. This is the place when the females' oocytes reside normally. Before they become oocytes, they are called follicles, guys. So these structures here, we're going to see this one, this one, this one, and this one are called follicles. They are not called oocytes. So primordial follicles, guys, let's say some words about them. Primordial follicles are actually the one which each female organism is born with. It's actually limited amount. I think primordial follicles are between 100 to 300,000 in both follicles, though. In both ovaries, sorry, in both ovaries. So they are limited in numbers compared to the males, which produce sperm all the time. You're going to see also here when you come eventually for the 15 practicum, you should be here. We're going to discuss this again. This is a very limited amount. So that's why female fertility has limitations, unfortunately, guys. Now, what's happened here? Primordial one, those are the one which the females are born. When they enter the puberty, guys, the primordial follicles we're going to see here starting to develop, so they become a primary follicle, and then become secondary, and then become a Graafian follicle. And in the end of the Graafian follicle cycle, guys, we have the ovulation with the oocytes leaving. The ovary goes in the fallopian tubes, and whatever left, guys, becomes a corpus luteum, which is the yellow body, which produces, we're going to see what when you come here on the week number 15. At the moment, we are just interested in the oocytes, guys, the one which are located inside that one, because they can be very big cells. You're going to see here up to 200 micrometers. I told you 150, but they can become up to the 200 micrometers. So here how it looks like, guys. This is the primary follicle, the one which is actually how it looks like after the initiation of the after the female already has the sex hormones, let's put it that way. So this is the primary follicle. How it looks like. Here is the schematic representation. Here is the live image. What you see here, guys, this big structure here is called oocyte. Outside of the oocyte, you have this pinkish color. This one I draw actually on the top of it, even though it's difficult for me. This one, which is called the zona pellucida, and outside you have several layers, like 1, 2, 3, 4 layers of cells, which are called follicular cells. And this is the primary oocyte, guys, primary oocyte. This primary oocyte develops further on and turns into the secondary oocyte. Here is the primary, you're going to see it, and then it turns to the secondary, which looks like this. This is the secondary, guys. Now, secondary oocytes, what is typical for it? It has the oocyte even bigger inside. Then you're going to see two different surfaces here. This is a surface, this is called a membrane here on the surface, guys, or you can also call it basal lamina here, on which you're going to see the separation of the internal part of the follicle with the external part of the follicle here. So internal part, you're going to see the oocyte, you're going to see some follicular cells, we're going to call them already granulosa cells here, you're going to see that one. And of course, you're going to see some holes like this one here, like this one here, like this one. Those holes, guys, are called anthriums. Anthriums are openings within the follicle cells, or actually they're achieved by dying follicle cells, which actually produce a liquid inside. So this is the follicular liquid what we're talking about. Now, outside of this membrane, you're going to see these dots here, is located the two thecas, the two sheets of the follicle, which we call theca interna and theca externa. The both of these theca structure, guys, this is the borders of the follicle here. The one I'm drawing you now. You see it? It's pretty big one. This is the inner part of the follicle. So everything else you're going to see here are the theca interna, theca externa. Those things are responsible for the production of the estrogen within the follicle, guys. Very important things to do. Now, what else we need to see? Well, we're going to observe now the final follicle, or it's called Graafian or mature follicle. I'm going to see Graafian mature follicle is actually pretty big one. This is the last stage of the follicle development, guys. Typical for it, you see the whole space is actually taken by the antrium, guys. Here only we have, in the middle of it, we have a small structure which is called corpus oophorus, guys, which holds the oocyte. This part here is the one which is actually released during ovulation, guys. This part here, this part. This part actually contains several different structures. Inside, in the middle, let me just zoom in a little bit. Here is the oocyte. You see it, this one. Then this line on top, this line here, this one is a zona pellucida. And outside, the one that's formed like a shadow outside, this is a corona radiata, guys. This is leftover of granular cells. Very important one to know. So here is the membrane, the one we talk about, which separates the inner of the outer structure of the follicle, guys. And then this is the other side, the theca interna and theca externa. I just draw them out. So when this is removed, what's left over is the outside of the follicle, plus some blood vessels, blood which goes inside during the rupturing. So these things forms actually the leftover of the follicle forms corpus luteum, which starting to express not estrogens anymore, but starts to express progesterone, guys. Okay, this is about the ovary, guys. We talk about it. What we see here by the description is the size of the ovary, of the oocyte. Here, which is pretty big one. We said here up to 200 micrometers. Now, we're going to continue next with the small intestine. Small intestine, guys, is a very interesting structure. Those are also, we call them guts. They're extension from the stomach. They start from the lower part of the stomach and continue on. You know that the small intestine is pretty large, one I think 3.5 meters around in the length, and then it turns into the colon afterwards. So small intestine, guys, how it looks like. Well, small intestine in generally, it has a very interesting structure. Here I give you a summary or a schematic representation of the small intestine. What do we see here? Small intestine is built of 4 different layers. Starting from this is the inside, but it's actually outside of the tissue. This is the outside of the guts. So this means when you hold it, this is the outside, but it's actually inside. So starting from the outside in, here, this way. So what you're going to see with you guys, this layer here, the one I draw on the top of it, guys, this is the epithelial layer, or called also lamina epithelialis. Lamina epithelialis, we're going to see with you, in it has 3 different layers. I forgot to tell you, the first layer is called tunica mucosa, of course. But lamina epithelialis has 3 different types of cells there. The most common population of them are called simple columnar resorptive epithelial cells. Those structures here, simple columnar resorptive epithelial cells, they're not ciliated. Don't look at this one, this is wrong. Now, underneath them, we're going to observe a basal lamina, this one, or lamina propria as highlighted here. Underneath that one is the muscularis mucosa. Here we have some muscle cells, smooth muscle cells. We have some loose connective tissue, guys, and we have a lot of blood vessels. A lot of blood vessels inside. So those are the one which feeds the epithelium, because epithelium is not blood supplied, guys. Very important to know. We're going to teach about that. After that, we're going to see talus submucosa, here this structure. Talus submucosa, guys, contains a lot of glands. It's a connective tissue, but contains a lot of glands inside. We're going to see them with you, don't worry about it. Underneath, that one is called tunica muscularis. Tunica muscularis in the guts, especially, it's formed of two layers, stratum circularis, stratum longitudinale. This one. So if this is your guts, guys, looking from this outside, also the villi, the one I actually talk about now, like this. So the muscle is the one is making the whole circle around the guts. The other one, though, it goes this way. So this one, when it contracts, actually shortens the size of these guts. And the other one, when it contracts, if it's like this, when this contracts, makes it something like a wave. So it pushes the food, which is actually inside, pushing it towards the exit, guys. That's the important part. This is called peristaltics of the guts, guys. And then finally, this structure here is called mesothelium. Mesothelium or mesothel, which is actually simple squamous epithelium on the bottom, and then we have the lamina propria of the whole epithelium. We're going to study with you that each epithelium, guys, has the basal lamina on which one is lying down. So this is in general the structure of the small intestine. So what you're going to observe with you guys is this. These are the villi. Do you see them? This one. You're going to see here lamina epithelialis. Then here somewhere is the basal membrane, guys, the one I draw. And this one which left over underneath that one, these structures here. This one, it's called muscularis mucosa. You're going to see smooth muscle cells, connective tissue cells, and other type of cells, blood vessels also inside. Very important one. Now, what we're looking with you guys today, it's about the cells. Here is the cell shape. Those are columnar epithelial cells. How they looks like? They looks like this. This is the basal membrane. They looks like that because they don't have enough space to spread around, guys. Very important one. Now, what you're looking here, guys, are the most common population here. This one with the elongated nucleus. So when you do a columnar epithelium, normally you do it like this, and then you draw the nucleus like that. Because it doesn't have space to make a circle, that's why it looks elongated one, guys. As it's perpendicular, I'm going to talk about this one when you get into the epithelial cells or epithelial tissue. But anyway, let's go on. So those are the most common population of cells. We call them simple columnar resorptive epithelial cells. Typical for them, guys, I'm going to zoom in a little bit here, is the presence of this line on the top. Do you see it? This line on the top, guys, it's called brush border. We call it brush border, or you actually call it microvilli. This one we're going to discuss probably, I don't know, maybe next week with you, or also we can start today to talk a little bit about it. Let's see how it goes. So those are the brush border, the microvilli. So this is one of the cells. You remember this one? I draw it kind of hard here. Another type of cells you're going to see is this one here. I purposely draw it here. This one, those are the stem cells. We look at the nucleus, it's more kind of circular one. And finally, this one here, I'm going to draw the outline of the cell here. This one is called a goblet cells. What you're going to notice that above stem cells and above goblet cells, you don't have this microvilli. They are typical only for the column resorptive epithelial, simple column resorptive epithelial cells, guys. Okay. This is what we are supposed to observe with you today here. Unfortunately, we should also see the muscles, but this one we will see when we get into the muscle tissue. Don't worry about it. Now, we continue on with the next slide. The next slide, we're starting with the neuronal tissues here. A lot we're going to talk about. So the first neuronal tissue we're going to observe, guys, is called medulla spinalis, or we also call it spinal cord. Spinal cord, guys, you're going to see here, it's built of two different types of matter. You have the white matter, which is here, and you have the gray matter, which is this. the gray matter. Gray matter normally have the shape of butterfly, as you know, or the letter H, but I prefer butterfly. So, gray matter, as we like to say, has two horns. This one is called posterior horn, this one is called anterior horn, or front one and back one. So what is typical for them? From anterior horn, guys, you're going to see this one comes the neurons which brings the signal for the receptors. So those are usually sensory neurons. They pass through some structure here, which is called ganglion spinale, guys. We're going to see it with you for sure. We're going to look for it. Now, this one are the most, not the important parts for today, guys. Important parts for today is located in the anterior horn. From here start cells which are multipolar. Then let's try to draw them like this, like stars shaped cells, guys, which actually are called motor neurons. Those are alpha, beta, gamma motor neurons, for example. Those executory neurons, which actually innervate the muscles and makes the muscle contraction, guys. Okay, so what we're going to see here, as I said before, is the star-shaped like cells. So this one we're going to see here. I'm going to surround it, the different stuff. Here, guys, what you're going to see, this is the white matter. White matter. This is the gray matter here. You see, the gray matter is stained more predominantly because it's formed from the bodies and the dendrites of the neurons. White matter, as you know, it's formed of the axons of the neurons. Therefore, here we're not going to see nucleus, or at least not the nucleus of the neurons, at least. But you're going to see the nucleus of the myelin-sheeted Schwann cells. Now, zooming in here, what you're going to observe with you guys, just give me a second just to change here to the hand in order to move it a little bit. Now, so what do we see here? So we see here that those are the nucleus of the neurons. Since we don't see the bodies because they're very densely packed one here, we can orientate according to the nucleus. So for example, this nucleus has several spikes. I'm going to draw them like here, like that, to see it, guys. These spikes, one of it for sure goes into the axon. For example, the thickest one, normally this one goes to the axon. So you're going to imagine there's a big axon going here, most likely going this way though. All right. The other spikes like this one, and that one, guys, are the one which goes inside the dendrites. Normally, when we have bipolar cells, it has only two spikes. So this is how the bipolar neuron should look like, guys. Like this. Here is the nucleus. So nucleus should be looking something like this, elongated nucleus like this. But this one has three spikes. This means that we're going to observe one axon and at least three different dendrites. This means that these cells are multipolar cells. This is how we're going to orientate yourself, that these are indeed the multipolar neurons, guys. Here. Now, we continue on with the next neuronal tissue, which is actually cerebellum. Cerebellum, or also called small brain. What is typical for it compared to the medulla spinalis, which we just saw, guys? Here, the gray matter is above the white matter. White matter is here down. This is the white matter. This is here the gray matter. You see, this is also written here on the pictures. White matter, it's below the gray matter. This is opposite to what we see into the spinal cord, guys. The gray matter is separated in three different parts. We have molecular layer, ganglionaric layer, and granular layer, guys. But important here to know not the name of the layers yet, but to know that this one in the middle, these cells are called Purkinje cells. Purkinje cells are very interesting. You're going to see them now. They look like pears. That's why we call them pear-shaped cells, pear-like shaped cells. This one here, this one here. Those are bipolar neurons, guys, which has one dendrite tree like this, and then one axon which comes from here like that. towards to the white matter, guys. They are pretty big one, very easy to spot. Do you see them here? Okay, then, well, let's continue on with the final slide for today, guys. This one is called telencephalon or cerebrum here. So, cerebrum, guys, is the higher brain. Higher brain. It has two hemispheres, as you know, and everything as a mental structure and stuff happens here. Now, cerebrum has 6 layers. You're going to see them 1, 2, 3, 4, 5, 6 layers of cells. They are separated. There's not only cells. There are actually 6 layers in which you have different types of cells. In the 5th layer, guys, this one here, we're going to observe a very big cells. Those are pyramidal neurons. Actually, all of them are pyramidal, but these are pretty big ones. Those are the big pyramidal neurons of bats. How they look like? Well, here it is. This is the layers, guys, here. For example, here is the top one, and then comes inside, and this is the 5th layer before the multiformis, and here is the white matter behind. So let me zoom in to show you how the pyramidal neurons looks like. Here they are, pyramidal. A little late. Трябва да изчакате малко, 12 тук. Okay, sorry for the interruption, guys. So this is the pyramidal neurons. You're going to see them. Here is the pyramid structure, guys. This is the axon, continuing in axon, and these are the two dendrites you're going to see, guys, like that. This is the pyramidal neurons for you. Okay? With that, I finish with the presentation. If you have some questions, guys, you can type them here. Maybe I can answer some of them. If not, I can wish you a good day, because I think, I don't know what time is it in your case, but in me it's almost lunchtime. I think this is enough for today. I will try to catch up next week with the other subjects. Don't worry about it. We're going to overlap sometimes. Okay. So if you have some questions, PowerPoint, it's only here. When you come here, then I will probably give you the copy of the PowerPoints. They are not inside the university accounts anywhere. Other questions? Okay. I think you do. Just wait a little bit to check who is here in order to don't miss someone, guys, because the presence is absolutely necessary to be here. So let me just check who is here. I'll just schedule it in my graphic, so I don't miss anyone by chance. And then I wish you a good evening. I guess it's evening in your case, right?
Yeah, good evening.
Okay. Okay, everything's fine. Good. And then, okay, so the guy, we didn't get the password. Usually they give you the password 1, 2, 3, I think, or something like this. I'm not completely sure, though. You can ask. Send an email to the guys who actually send you the email, so you can actually talk to them. Give them a feedback that you didn't get it, so they will send you another password. Yeah, sure. This is actually very important to see the slides for yourself. But unfortunately, since we are not yet here, we will need to do it this way. Otherwise, afterwards, I will give you the time to see all the slides, guys. Don't worry about it. Okay. See you next time. Next week, we will talk a little bit about cell membrane and the structures on the membrane and the plasmic reticulum, ribosome. So if you want to read about this one, guys, don't worry. Okay. See you next time.